Just going to say a quick hello to those of you who have joined us so far. Thank you and welcome. We're still waiting on several people here, so we'll give it a few minutes. We'll probably start a couple minutes after 3 p.m. Eastern, just in case we have a few stragglers. But thank you and welcome. You're in the right place, and we'll be started in less than 10 minutes. For those of you who've just joined us, thank you and welcome. We're still going to wait just a few minutes. I think some people may only be hopping on now as it's turned 3 p.m. So we'll wait a few minutes, but you're in the right place. Thank you for being here, and we'll get started in about 4 or 5 minutes here. All right, I have 3:03, so I think in the interest of respecting everybody's time here, we'll get started. So hello, good afternoon, good morning, good evening, welcome to the first Canadian Grapevine Certification Network webinar of 2022. Our focus for this session here is on grapevine trunk diseases in mature vineyards. This is a follow-up webinar session to one we ran last year. My name is Ethan Churchill. I'm the project manager for CGCN, and I'll be serving as your moderator today. Also here with me representing CGCN is Bill Armstrong. He is on our board of directors, and the two of us sit on CGCN's Knowledge and Technology Transfer Committee, or KTT committee. So we're responsible for sharing knowledge with industry and hosting events such as this one today. We're also very fortunate to have two very distinguished guests with us here today. We have Dr. José Ramón Urbez-Torres, and we have Dr. Mark Siznowski, both of whom will be talking about grapevine trunk diseases in Canada and Australia, respectively. We'll also hold a question and answer period near the end of the webinar, so feel free to use the Q&A feature you should see at the bottom of your screen there. You can type questions in at any point, and at the end when we have our Q&A session, you'll be able to have those read out by Bill, who will be moderating that for us. Make sure if you're using the Q&A as well, please just mention who your question is directed towards, just to make it a little bit easier for Bill to moderate. I'll let everybody know as well that this webinar session is being recorded, and it'll be available on both the CGCN English and French websites in the next couple of days or so, as well as on our social media platforms, which you see on the screen there, our Facebook and Twitter accounts. So if you ever want to go back and review this or send the link to somebody who wasn't able to make it today, that link will be posted in the next coming days or so. Before we dive in here to José's presentation, I'll just give you a quick brief presentation on CGCN, tell you a little bit more about us as an organization. So let me just pull up my right screen here and we'll get started. All right, this is working okay. Everyone can see the proper screen. Awesome, seeing a couple nods, fantastic. All right, so the Canadian Grapevine Certification Network was incorporated in February 2017, and it's comprised of membership from the 4 grape growing associations in Canada. So we have the BC Wine Grape Council, the Grape Growers of Ontario, the Conseil de Vin de Québec, or CVQ, and the Grape Growers Association of Nova Scotia. Each of those member associations have 2 elected members to the CGCN Board of Directors. The CGCN board has maintained a mandate that is really twofold. Oh, there we go, sorry about that. Firstly, the main goal here is to advance the Canadian grape and wine industry by ensuring a sustainable domestic supply of certified propagative grapevine material, which includes both rootstock and scion wood. Particularly viruses of concern for the industry at this time are leaf roll and red blotch, which result in significant economic losses if they're left unaddressed in a vineyard. Second main part of what we do at CGCN is being the lead for the Canadian Grape and Wine Science Cluster, which I'll talk about in a little more detail right now. So the application for the Agroscience Cluster was submitted back in January 2018. All the cluster activities were built on past research in each province in order to create a national coordinated effort to address some key challenges in the grape and wine sector. Our application for the cluster was approved in May 2018 for $8.4 million in federal funding through the Agriculture and Agri-Food Canada's, or AAFC, Canadian Agricultural Partnership, as well as $3 million in industry funding over 5 years for 23 different research activities. There's just over a year remaining in this program right now, ending in March 2023. The cluster activities, as I mentioned, built on research from past provinces to have a coordinated effort across the country. And those research activities are broken down into a few key themes, which are listed here. So firstly, we have strategic management of grapevine virus diseases. We have cold hardiness and climate change, sustainable management of soil, water, and crop quality, optimizing the quality of Canadian wines, crop protection and monitoring, and lastly, knowledge and technology transfer, which for us is a big key of the whole piece in terms of providing information and technology back to the industry. Part of that, of course, includes the webinar that you're all here for right now, But other KTT efforts include the development of our websites in English and French to share a lot of this information directly with the industry and with the public, use of our social media channels, and pre-COVID, some research events, as well as scientific publications, a regular newsletter we post, and much, much more. So KTT is certainly a big part of what we try to do as part of the cluster. Here you see a snapshot of our website, because research updates from the cluster are posted there as they become available under the research heading at the top. We also post on a pretty regular basis, most recently a number of cold hardiness updates, as well as growing degree updates for BC, Quebec, and Nova Scotia, as well as plain language summaries for each of the research activities, and those are usually released on an annual basis. But I would encourage you, if you're curious, to check back on our website regularly, as there are pretty regular updates to research and new information being added. There you go, a little circle at the top for the research button there. So now I'll move on to talk about the other half of the CGCN mandate, which is our Clean Plant Network. In June of 2019, more than $2.3 million in funding was announced through the Canadian Agricultural Partnerships Agri-Assurance Program in order to create a network of certified virus-free grapevines that Canadian grape growers can plant in their vineyards, which helps to ensure the long-term viability of the Canadian grape wine sector. This funding is allocated to our interim verification standards and our long-term certification program, and I'll briefly touch on both of those right now for you. So these two clean plant protocols we run in parallel for the time being. The certification program contains two different subsets of protocols. Option one is the most rigorous. It centers around propagation from CGCN's Generation 1 or G1 repository, which is currently tested and held clean at the CFIA Center for Plant Health in British Columbia, along with help of nurseries through the Generation 1A, G2, G3, and G4 levels for grapevine material that is tested and certified from a whole host of viruses before being sold to grape growers at the G4 level. The option two protocols, which was initiated in early 2021, allows nurseries to propagate material from pre-existing vineyard blocks at the G3 level that have undergone thorough testing using high throughput sequencing or HTS. And while it's not quite as rigorous as the option one protocols, option two still allows nurseries to provide certified grapevines to buyers in a very timely manner without compromising on thorough virus testing. The interim verification program was put in place to serve as a temporary option for virus tested grapevines until there was a sufficient stock of certified material available under the long-term program. And this verification program works with nurseries to test existing propagation blocks for leaf roll 1 and 3, red blotch, and Pinot Gris virus. Testing for this is offered at a 50-50 cost share between the grower or winery and CGCN. And we work with both nurseries for general propagation, as well as wineries and grape growers who wish to do custom propagation of their own vineyards for this program. We also, as part of this, encourage growers and wineries to contact us if they wish to see certain varietals available through the programs. And we're consistently monitoring the industry to see what's wanted and ensure that our repository is reflective of those needs. So if ever there is a wish list item from you, you can submit that through our contact page on our website. I think there's, a little circle at the top right there. Or you can email the contact at the end of my slides here, and you're always welcome to submit information through that as well. And that is the contact information I was speaking about. So that's my contact info there, my email, phone, and then the link to the CGCN website as well. Thank you for allowing me to talk about our program for a couple minutes here. I will certainly answer any questions if any pop up in the Q&A, or if you think of them later, by all means, or you can reach out to me via email as well. So I will stop sharing my screen for now, and then I'll introduce our first speaker. Okay, perfect. All right, so I will now introduce our first guest speaker. We have Dr. José Ramón Urbez-Torres, who is a research scientist at the Agriculture and Agri-Food Canada Summerland Research and Development Center in British Columbia. He also serves as adjunct professor in the biology department at the University of British Columbia, Okanagan campus. Jose received a postgraduate master's degree in viticulture, enology, and wine marketing in 2001 from the International Social Science Council, and a degree of agricultural engineering in 2004 from the University of Valladolid in Spain. He completed a PhD in plant pathology in 2009 at the University of California, Davis. Dr. Urbez-Torres has studied diseases of woody perennial crops since 1999, and his current research at AAFC focuses on the development and implementation of sustainable management strategies against fungal, bacterial, and viral diseases of grapevines and tree fruits in Canada. As of 2021, Jose is also a professional affiliate at the Cool Climate Enology and Viticulture Institute, or COVI, at Brock University in St. Catharines. Without further ado, here is Jose. Oh, you're still muted there, Jose. Are you able to turn the microphone on?
Oh my God! Of course, that wouldn't be a meeting without this. I'm gonna try again. Hold on a second.
Take your time. Thank you.
Yeah. Okay. Can you see the presentation now?
Not yet, no.
Is it coming? No?
Still nothing yet, no.
Okay, let's try again. I'm gonna disconnect this. Maybe that's the problem. Well, this is what happened with the virtual. Okay, let's see.
Okay, yeah, it's loading now.
Can you see presentation now?
Yeah, that's perfect. Thank you.
Okay, finally. I don't know. Anyhow, okay. Well, thank you very much for the introduction, Ethan, and thank you very much once more for inviting me to the CGCN webinar series to present part of the research we are doing, as you mentioned, within the program, the CAP program and the funding we received from CGCN, the BC Agriculture Council, and Agriculture and Agri-Food Canada. And today I will be talking about managing grapevine trunk diseases in mature vineyards. And this is a continuation of a webinar we had last year, talking into managing trunk diseases in young vineyards. So today I will present a very brief introduction just to set the stage on the problem we are going to be talking about. And then I will be focused on the work we have been doing in developing and implementing some of the chemical, biological, and cultural practices here in Canada, as well as ending with some of the future directions and steps that we would like to continue taking further. So without no more ado, I would like to introduce, many of you already are familiar, but what grapevine trunk diseases are. So these diseases are caused by fungal pathogens, in this case by many different fungi. These fungi can be either in the Ascomycota or Basidiomycota. Most of these fungi belongs to the Ascomycetes. And these pathogens infect grapes through any type of wound or opening in the plant. This is the main infection that happens in the vine. As we know, we prune vines every year to be able to get a crop. So as you can imagine, pruning wounds are the main point of infection. However, other type of wounds, mechanical damage, retraining, are also points of entry for the pathogen. Some of these fungi are thought also to be latent pathogens, and we talk about that more in the previous webinar on the situation on the young vines. But basically what this means is they may be part of the vine and not causing any problem. And only when the vine is under certain type of stress, they transition in from this endophytic phase where they don't cause any problem to pathogenic phase. The overall symptoms include a slow or rapid dieback, and that will depend on the disease or fungi involved. And as you can see here, some of these cordon dieback, progressive death of the plant parts, and of course, eventual death of the vine. The distribution of these diseases is worldwide. Wherever we grow grapes, you will see these diseases occurring. However, it's important to mention that the different diseases and even the pathogens involved can vary depending on the geographical region. We have two main groups of trunk diseases, whether we want to call them as a disease complex or complex diseases. First, we have in young vineyards, during the first years of establishment, we have blackfoot and Petri disease caused by a species within this group of genera of fungi. And overall, they cause what we know as the young vine decline, causing, as I mentioned, usually dieback and mortality of the plants in the first years after establishment. Then we have the diseases in mature vineyards, we will be focusing today, of course, and those include ESCA, which is caused by pathogens within the Petri disease complex, as well as some of the basidiomycetes, and the canker diseases, Botrytis dieback, Eutypa dieback, and Phomosys dieback. Usually, you start seeing symptoms of these diseases when vineyards are 5 years, 8 years of old. So today we are going to be focused on mature vineyards, as I mentioned. So we have ESCA disease. As you can see here, some of the symptoms we can observe inside, internally in the bud. Very characteristic of ESCA is this yellowish, spongy, softened bud that is caused by the basidiomycetes involved, which is not usually seen in the canker diseases. We have very typical characteristic foliar symptoms as the tiger stripes, the sudden collapse or apoplexy that we see in the vines, or the disease can also cause what we know as black measles, which is infection or symptoms in the fruit. However, today we will mostly focus on canker diseases. As I have to mention that ESCA is not very prevalent in Canada for what we know, and we have a study. However, it's the main disease that European growers are dealing with. We have canker diseases, as you can see very clearly. This is the main symptoms we are going to observe in the vascular tissue. These are the web shape, or sometimes can have a different form, these cankers. And the two main diseases are Eutypa dieback, and here I include some of the symptoms that this disease will cause these foliar symptoms. As you can see, these short internodes, this cupping of the leaves, as well as very dramatic impact on the fruit. And then we have Botrytis and Phomosys dieback, which we can differentiate mostly because these diseases, we are not going to see a very clear foliar symptoms. We have either what we call these, dead arms or blind cordons with no foliar symptoms, where the spurs, are not having any spring growth. Or in some cases with Botrytis dieback, we can see also the canker and also normal growth on the plant. So that's one of the ways we can differentiate the two main diseases. Regarding the significance of these diseases and why it's important, we are studying them and looking for management strategies. These are studies conducted by Jonathan Kaplan at the University of California. And we can see here, we have the vineyard age and the incidence of the disease. And of course, as the age of the vineyard increase, we are gonna have the increase on the symptoms. Usually, we start pruning severely the vines after the 5th, 6th year, more pruning was available, infection of the spores, and these symptoms are going to significantly increase. At the same time, the symptoms increase, we are going to have parts of our vines dead, spores without production. So we are going to see, of course, the yield decreasing. After 10 years, we can see a 20% yield reduction, but between 10 and 15 years, depending the disease, the geographical region, you can see up to a 50% decrease on your yield, which, as you can imagine, causes significant impact. Just briefly, the epidemiology and how these diseases work. We talked in the previous webinar how one of the sources of inoculum can be the planting material, where these pathogens can be introduced into the vineyard. But in the case of mature vineyards, we are going to have, from infected vines, we are going to have these fruiting bodies with the spores inside that under optimal environmental conditions, the spores are going to be released. We are going to have infection in our pruning guns, canker development starts, and the cycle continues. These spores can not only come from our vineyard, from infected vines, but also many other different hosts. In this case, in the Canada, we have done studies with cherry and apple are some of the main hosts that can also provide inoculum to vineyards, as well as native plant communities, and this has been widely studied. So this fungi has a very wide host strain, and not only grapes, but other hosts can be involved. In terms of the control where we are right now, resistant cultivars, my colleague Dr. Mark Siznowski will talk later about this, but as of now, all vitis vinifera is susceptible to trunk diseases. We have seen differences in susceptibility, but all vitis vinifera is susceptible, and there have been studies where even wild vitis species or hybrids are also susceptible. So we don't have really that option. Some of the products that have been effective in the past have been already banned or phased out due to concerns into human, environmental, or animal health, so they are no longer available. We currently don't have a product to control trunk diseases when the plant is infected. So when the pathogen is inside and the canker is forming, we don't have any product there. And we have to rely on remedial surgery that we will describe later, is the only way we have right now to eliminate infected parts of the vine. So our current control studies are primarily focused on preventative measures, and that's what we are going to be mostly talking today, which are pruning gum protection, as well as cultural practices that we can conduct in the vineyard to minimize the infection. So moving to the next, the work we have been doing here, in trying to start developing these chemical, biological, cultural practices in Canada. First, I would like to talk that contrary to other grape growing regions or countries, trunk diseases have been recently discovered, not that they just suddenly appear. They have been here in Canada for sure, much longer time, but it was basically around 2008, 2009, where my predecessor, Dr. Peter Schorber in BC, started to conduct some studies on trunk diseases, because growers started to see some of these dramatic impact, on vine mortality. The studies we have conducted in British Columbia show that in mature vineyards, we can have up to 80% grapevine trunk disease incidence in vineyards, and you can imagine the impact that may have in yield and production. And then we can see here the incidence versus vineyard age. We have basically two main groups. What we called before in the previous seminar, the young vine declines, and vineyards with very high incidence of this young vine decline. And as disease increase, we can see also how the disease incidence increase significantly. Most of these vineyards, 15, 25 years of age, with up to 50% incidence, other vineyards with much higher incidence. So we have been studying the biology and epidemiology, and we have significantly made breakthrough in understanding these diseases here in Canada, in British Columbia. But one of the things I have to mention compared to other countries is we currently don't have any product register. So there are no products that a grower can actually start using to control this disease. So a lot of our focus has been on generating data for product registration, as well as implementing control strategies under busy conditions. So our first approach I'm going to start was to start in vitro screening of chemical and biological products. And this is a project we started in 2016. Just the rationale behind all these different treatments we have here and active ingredients was, first, we were looking for products that they were registered in Canada and in grapes as well, with the idea that if these products were successful on controlling trunk diseases, we could have a label extension of the product, which is much easier than have a full registration from scratch. So, for example, products like fipronil, plasfredoxinil, or primethanil, used for control of powdery mildew. The other idea was to have products that they are registered in Canada in other crops, but not in grapes. But again, if they are successful, to have some label extension towards trunk diseases. And of course, some products that they are not registered in Canada, but we know that they have been shown promising and very successful results in other countries. So we wanted to generate some data under our conditions in order to see if the registrants would be interested in bringing these products to Canada. So as you can see, a very large number of products we started. And in order to do that in a high throughput and in, I would say, easy way, we developed what we call first an in vitro screening in the greenhouse of detached KNSAs, so these DCAs. This I learned from my colleague, Mark Siznowski, and the group there in Australia. I was lucky enough while I was visiting Australia for the Trunk Disease conference to spend time with him. And these are results of some of the good collaborations we have had. I was able to learn how they were developing these detached KNSAs. And we have right now implemented and modified a little bit to our project. So basically what we have here, that this method is allowing us to have many different canes to be able to have a high throughput experiment. These are our greenhouse in Summerland. These canes are put in this styrofoam floating trays in water. So this allows us to have these canes for up to 6 months. They will start even rotting at the bottom. So it's a way that we can have these type of experiments because of the amount of products and pathogens. So we have a fresh pruning gun made where we then will treat the pruning gun with the product, and then we challenge these pruning guns with our different pathogens and doing the assessment. So we have a mean % disease control, and that's how we assess, which is basically comparing the recovery from our controls, non-treated, but inoculated versus the treated and inoculated. So just a snapshot of this first experiment we had. I'm showing here all the different products with all the different pathogens we tested. You have to see why I'm talking about this high throughput. We have up to 10 different species with many different products. So this system allows us to conduct that. And I'm showing here only those products that show more than 60% control in our trial. So you can see, and this is one of the problems we have with trunk diseases, it's very difficult to find a silver bullet, just a product that is gonna perform well with all the different pathogens. All these pathogens are taxonomically different. They have a different, probably infection way. So that's one of the challenges we have with trunk diseases. However, in this case, we observed that there were 3 main products, thiophanate methyl, and then tebuconazole-based products that they perform significantly well with high disease control in a large number of different pathogens. Also, we had to take into account that some of these active ingredients probably have underperformed due to the high inoculum. We were using 5,000 spores of the pathogen per boon, which is much higher than what will happen in natural conditions. So we have also to take this into account. But this served for us to select some of these products that they were performing well and to move to the next step. We choose also Trichoderma, and I will mention this why if we only, for example, saw 70% infection control here. These assessments are done when inoculations are done 24 hours after the treatment. And we know that with biological control of Trichoderma, we need to get the Trichoderma established in the pruning gun, and it's gonna give us a much higher control maybe after 2, 3, or 4 days, but not immediately after. But we select Trichoderma also, as I will mention later, because of the specific interest we have here. So as I mentioned before, those were results we get it when the spore just is inoculated 24 hours after treatment. But as we know, and these are results for our epidemiological studies when we were doing a spore trapping, where basically is telling us, throughout 3 years, all 3 growing seasons, we were able to collect spores and using molecular tools, quantify the total amount of spores we have in the vineyards with the idea to understand when the inoculum is present here in British Columbia. We can see that at the end of winter, early spring, it would be late March, early April, that's when we have the first significant amount of spores. And we see that these spores continuing being in our vineyards through the growing season, with the mass highest incidence, I would say until end of June, mid-July. So we have a period there of 2 or 3 months where inoculum can be present in our vineyard. Inoculation is also present later, but by this time, as I will show later, our pruning wounds are much less susceptible. They have been healing through the growing season. So basically, our main target would be these 3 first months, after we have the first catch. So because of this reason, we also need to know how long these products are going to be able to be in our pruning wounds. So for this case, we have a similar detached KNSA. And what we did here is we challenged the wounds not only 24 hours later, but we came back 7 days and 21 days to see how some of these products were efficient over time. So in this case, I'm showing results for 2 of our products, gel seal and green seal, tebuconazole base, that performed very well in our first DCA. And here you can see in black is our control. So this is our infection. These are our non-treated but inoculated. But to simplify, the visualization, what you have to pay attention here are the red, green, and blue are our total control, 24 hours, 7 days, and 21 days after treatment. So we can see here the first thing, as we knew, the product is not going to perform the same depending on the different pathogens, and that's a problem we know we have. However, we can see that the tebuconazole-based products, usually 24 hours later, even 7 days after, they have a pretty high mean percent disease control, over 70, 80%. But overall, we can see that after 7 days, towards 21 days, the efficacy decreased. And this is expected for many of the chemicals that we use on pruning wound protection. If we compare this, for example, with our biological controls, the situation is a bit different. As I mentioned before, we need our Trichoderma to get established into the pruning wound. Trichoderma, sorry if I didn't mention, for those of you who know, is very commonly used biocontrol fungus, and basically the main mode of action is, by invading all this tissue and all this pruning wound, will avoid for the pathogen to come and colonize. So as we can see here, we don't have such a success with, for example, with this product 24 hours later. Probably Trichoderma has been well established, but 7 days and even 21 days, when our pruning wound is well colonized, we are having a much better result. This case with other species, Trichoderma tuberide, we can see how we have significantly increased our control even 24 hours after treatment. So in this case, over 80% in Botrytis species, not so much with Eutype palata, but 7 days and 21 days later, the product has performed pretty well. Trichoderma most likely has colonized the entire pruning wound and is giving us very, very good control. So in this part of our work, we can see that we were able to generate, data for product registration. We are hoping that some of these registrants right now are interested in bringing these products to Canada. I know that some of them are in the pipeline to get registered here in Canada, so that's very good news. So thanks to this work, we are hoping to have some products in the market. We just got approved a product by PMRA, which is our pest management regulatory agency, that they are looking to have a label extension of thiophanate that we have proved that is a very good product with a wide range of control for many different of these fungi. And in this case, I will be working with Dave Neal in minor use program in Summerland, developing trials and generating data. So now I'm moving into the biological control for these diseases. And why biological control? Well, in our case here, it has been a significant increase in BC of vineyards farmed organically, about probably 20% of them. It has been a lot of interest, on sustainable grape production and developing IPM practices. We have more and more consumer demands, for sustainable production. So the industry has clearly made a priority the need of biological alternatives. So in 2018, as part of this CAP funding, I have a master's student, Dix Pollard, who the main work was focused on identify the Trichoderma species we have here in British Columbia in vineyards, and study how they will perform as biological control agents. So our first results among all 29 isolates, we were able to isolate from vineyards in BC. We observed that we were able to identify 7 different species of Trichoderma. And we do that based on molecular identification. This is a phylogenetic tree of DNA sequences that we are able to identify all the different species we found in British Columbia, as well as morphological studies of these species. So this brought us to have 7 different potential species that may have, good control activity. So our next step is to take all these 29 isolates and conduct what we said is a dual culture antagonism assay. And this is our first step to identify the best performing isolates. So basically in the lab, in vitro, we compare the growth of the pathogen in the presence of the biological control, the Trichoderma, and we compare with the control, which is, just the pathogen. So basically we are measuring the percentage of, inhibition of the mycelium that the Trichoderma can have. So in this case here, we have the mean percent inhibition for 2 of the main pathogens in the Botrytis. And we can see that all the isolates basically give us more than 50%. However, isolates within the Harcianum group or the Atroviride group give us the highest control, with over 75% inhibition. So this helps us to identify isolates, but again, you can see how different species, have different impact in controlling. In this case, Harcianum was much better for Parvum, and Atroviride was much better for Sheriata. So again, difficult to find a silver bullet for all the pathogens. So our next steps was to develop these detached Ken assays, as I explained before. So in this case, with all our best isolates, we were selecting, and here we have the disease control, 24 hours, 7 days, and 21 days. Same, we have seen that not all the species have a good control for both pathogens, but we were able to identify a specific species, that actually allow us, to move forward. In this case, with, close to more than 70 or even higher control 24 hours later, but importantly, they maintain a very good control for up to 21 days, with more than 80 or 90% control in this case for this species. So also what we do is, in this case, we know that in some of these, we can increase our control by having mixed inoculation of these best performing isolates. And in this case, what we did is those best species and isolates we found, we basically have mix, of the species, and we can see how significantly we increase our mean percent disease control for both pathogens, including 24 hours after. So if I give you before the results for the single species 24 hours, we can see how not all perform well. When we have a mixed inoculation of the species, even 24 hours later, we are able to find more than 90% control. So these results were very, very promising. And we were actually very excited because this is normally not what you would expect with Trichoderma. You still would expect that it would take more time to colonize. I had to say that these were control studies in the greenhouse. So we, of course, wanted to move that into the field. So for that, we have a 2-year trial in Merlot vineyard in Summerland. We prune these vines, 4 to 5 bats. And we use these 3 different Trichoderma-based products that we selected from our greenhouse. And we compare with the standards that we know they were working very good, the tebuconazole-based, as well as another biological control. In this case, we reduced the inoculum to 2,000 spores. So much lower inoculum pressure, still higher than what we would expect in nature, but to give a chance also for these products to perform better under natural conditions. And we screen how well these products were performing in the pruning wounds all the way up to 60 days post-treatment. So our first results for 2019, we have here, this is our control. So these are the non-treated but inoculated. And the first thing we can see is 24 hours after we prune and we inoculate, of course, the highest infection, the pruning wound is highly susceptible, but we know that the pruning wound susceptibility decreased, with the length of time. So still pruning wounds were susceptible 60 days after we prune them, for these different pathogens, but as you can see, susceptibility significantly decreased. We can see here in our mean percent disease control, and we were significantly excited about that. Even 24 hours after pruning, we have a very high protection. And compared to our standards, even the chemical, we have a very similar or better control with our biological control. We were able to provide a very good control over time, in this case with this last mix for up to 60 days. The pruning gums were very, very protected. Very similar results with Neophocyticum parvum, as we can see, very high protection from 24 to 60 days throughout the duration, and our biological control really compared to the commercial standards. I will go quickly, but just to show you the second year, again, very similar results. We have a much lower infection compared to the first year, and I believe is that the case that our inoculations were done much later. But again, we have the same trend, with pruning gums being highly susceptible just after and decreasing with time. Very similar results. We are happy that we were able to repeat these results on time in the second year. Very good control, not only 24 hours after, but increasing with up to 60 days. And again, even with the highest virulent pathogen, we were able to show how our biological control were significantly working and compared to standards. So moving into the next step, we have been doing also some work on cultural practices, and in this case, managing pruning time to reduce infection. And we wanted to see how different pruning times will allow us to minimize the infection for trunk diseases. So we know our inoculum is mostly happening in our conditions in March and April. So in this experiment what we did, we prune at different times, pruning in December, January, February, March, and April. And in this case, in April 16, we artificially inoculate all these pruning gums, and we observed that early pruning versus late pruning, we have a significant chance to diminish the infection. We have to 80% less infection in January. We were expecting to have much less infection in December just because the pruning gum has been longer exposed to healing. However, I cannot really explain why in December we got a higher infection. But overall, we see that pruning earlier versus late, we are able to reduce infection. If you are pruning in March or April, which many growers in this case will do, knowing that we have the inoculum coming during that time, those pruning wounds are highly susceptible and you have a much higher rate of infection. And finally, just to finish my part of the presentation, we have been also working on cultural practices, and this is a lot of things I learned from my colleague Mark, where they are experts and they have been doing that for a very long time in Australia, on how we could implement, remedial surgery, which basically the idea is to remove, the infected parts, until you are able to find a healthy boot where you remove all the infection and you are able to retrain and reform those vines. So in our case, we can conduct field trials, but we have a different aspect in the sense that in our trial, we observed that we were not able, the infection was already to the bottom. We have here the average in the 3 different varieties. Average infection, canker area on the top are in the bottom. Of course, higher in the top because that's where infection started, moving towards down. But even at the bottom, we found a very high infection. So these vines, they are infected all the way down. So our question was basically determine the number of production years left in these vines and the vine survival, because of course the grower couldn't afford to pull out, all the entire vineyard. So we wanted to see if this type of work will help the grower to at least gain time, to replace this vineyard. So in our results, I'm showing Pinot Noir and Pinot Gris. So applied in November 2016, we have that up to date, 100% of the vines have survived. We have that in our cut vines, the following year after cutting, we already obtained yield. That's why it's very important to plan ahead if you decide to do the remedial surgery and start deciding which type of cane you are going to be leaving. In this case, we see that the yield significantly decreased in the cut vines, in both varieties over the years. We can see how this yield is decreasing. And we can see how overall, after 2 years, our yield in the treated vines were significantly increasing, and in most cases, surpassing what we have from the uncut vine. So we were able to really demonstrate that it can be a good tool here to maintain more years your vineyard, to gain time, even if the infection is at the bottom of your vine. So just as a conclusion, we have chemical and biological control that we have been able to develop and implement. We are going to continue, of course, with data generation and continue with local. We have locally sourced biological control identified, as well as we know that we can work with the pruning time or remedial strategy to minimize this infection. In the sense of the moving forward, well, we need to really understand and determine optimum times for pruning and protection under our conditions, the survival of these biological controls under our cold conditions. Can we inoculate much earlier those vines? Search for other local sources of biocontrol, the effect that these biocontrols may have also on plant health. I think this is very important to study, and the effect of some of the extreme environmental conditions on our controls. Continue with data generation. We are very interested in implementation of some of these controls at the nursery propagation process. And the idea is continue all this work to develop a strong IPM program for plant disease. So with that, I believe I go within my time, and that's some of the resources of all the work we have been publishing, and is available, for you with all these DOIs, as well as some of the resources in books and other research letters we have with way more information I provide here. So with this, I stop sharing. I thank you very much, and I will pass to Mark. Thank you.
Perfect. Thank you, Jose. That was absolutely fantastic. I'll let Mark get himself set up, and in the meantime, I'll just do a brief introduction for him here. So Dr. Mark Siznowski leads the Horticulture Pathology sub-program at the South Australian Research and Development Institute, and is also an affiliate senior lecturer at the University of Adelaide. Over the past 20 years, he's been responsible for research on managing Eutypa and Batriosphaeria dieback diseases in grapevines and almonds in both Australia and New Zealand. He is the Australasian Regional Representative on the International Council for Grapevine Trunk Diseases, and he was the chair from 2014 to 2017. Dr. Siznowski draws from his extensive research experience and international collaboration to provide industry with the latest information for effective management of grapevine diseases. Without further ado, here is Mark. Mark, as well, if you want to switch your display setting like last time so we can see your the full screen.
Okay, I thought I'd already done that.
Yeah, there you go, perfect.
Is that better?
Yes, that's it. Thank you.
Excellent. Thank you very much, Ethan, and thank you to CGCN for inviting me to present on this occasion and to talk about the work that we've been doing in Australia and New Zealand on grapevine trunk disease management. As Jose has pointed out, we work very closely together with other colleagues in Europe as well. So in Australia, we're not too dissimilar to Canada, where we deal mainly with the two diseases, Eutypa and Batriosphaeria dieback. Jose's done a really nice job of introducing those diseases. It's not to say we don't have present the pathogens that cause some of the other diseases. We certainly have levels of Phomopsis dieback, and we have many of the organisms responsible for ESCA, but we don't very often at all see the symptoms of those diseases. So Eutypa and Batriosphaeria are the diseases that we've been spending a lot of time working with. Jose also talked about the disease cycle, so I won't talk about it into any more detail, except to say that we obviously, in trying to manage these diseases, there's two areas that you can really make a difference, and that's one, to stop the disease, the spores getting into the wounds in the first place, which is obviously the ideal scenario and what we've spent most of our time working with. And then when we already find ourselves with infected vines, getting rid of that infection is the only other control strategy that's really shown any efficacy. So varietal susceptibility, Jose also mentioned that all varieties are susceptible to the disease, and that's true. We can successfully infect any variety that we try. But what you can see here is a bit of a gradation in the amount of susceptibility in different varieties. This is a germplasm collection that we have in the Barossa Valley in South Australia. These vines are in their 30s year old, so they're quite mature vines. And what you can see here is the brown bars on the left showing you the amount, the severity of dieback, and in the green bars, that's just looking at the foliar symptoms of Utica dieback alone. So really concentrating on the dieback, you can see that there's many varieties that are very susceptible to dying back from trunk disease. And as you go across to the right, you see there are varieties which we really struggled to see any symptoms in the few years that we assessed these vines. I've highlighted here the varieties that are predominantly grown in Australia and New Zealand. And as you can see, we do have a predominance of varieties here in Australia, such as Cabernet Sauvignon and Shiraz, which are very susceptible. And in New Zealand, their number one variety, which their industry really hinges on, Sauvignon Blanc, is probably one of the most susceptible varieties that we've come across. So it really does mean we have a great challenge ahead. Just for the purposes of this talk, I've just highlighted some of the more commonly grown varieties in Canada. And obviously you are presented with a different challenge than we are, even though the varieties here in these conditions, because thinking about this germplasm collection, it is in Australia and with Australian isolates or Australian species of the fungus. You know, the amount of susceptibility is not anywhere near what we have to deal with in Australia and New Zealand. So, but even given that, as Jose's shown, problems still are arising for you guys. So just some interesting work that we've been doing with susceptibility. We did take it a step further because we thought there was some really promising material or varieties that had very low levels of symptoms in the mature vines. So we actually went into the vineyard and we inoculated canes, a bit like what Jose has already described. We also used our detached cane assays. And so these two scatter plots here show the two different methods that we used and I guess the recovery distance that we got of the pathogens into the wood. And here you can see the green circles for both Utica and Botrytis dieback pathogens, just highlighting some of the varieties that had clearly a lot lower level of disease progression. So there was some sort of reaction by the plant to slow down the progress of the disease, which really is a form of tolerance to the disease. So incidence of the disease in Australia from a fairly major survey we did many years ago, you see a similar pattern to what we saw Jose show for Canada. We obviously have a lot more mature vines in up to 50 and plus years that we could access for this survey. But the interesting things to see here is that the incidence of disease, as early as 5 to 7 years, we start to see blocks showing dieback. Now, this is clearly dieback that's come from infection in the wounds, not from propagation material. So it's not young vine disease, it's actually the mature form of the disease. But you can see here this infection that's happening so early in the life of the vine. We know that the symptoms from the time of infection to the symptoms showing and to dying back of spurs is at least 5 to 8 years. So we can assume that these infections that we see at this time are happening right at the beginning of the vine's life. And I'll talk a bit more about that a bit later. As time goes, you can see between 10 and 20 years, this explosion of symptoms in vineyards up to 100% in some cases. So it's by the time vines are 20 years of age in Australia, most growers are very acutely aware of the amount of disease that they see. But keeping in mind, it's not all doom and gloom. You can have mature vines with low level of disease. One thing that obviously helps is the variety. Having a more tolerant variety means that infection moves slower and allows you to have a longer sustainability of the vineyard. But even with some of these more susceptible varieties, there's obviously promise in using management strategies such as particularly remedial work to keep the level of disease to a much more manageable level. In New Zealand, we also have done a lot of surveying, and this graph here is a similar scatter plot that you can see of incidence by age. Obviously much younger industry, very similar to what you have in Canada. And what's really interesting in New Zealand is the difference between the three regions here that I'm showing. And the main reason for that difference is, sorry, I've been pointing at the wrong screen, I think. So the main reason for the difference is climate, and what we know about Hawke's Bay is it has a much higher rainfall, and so does Marlborough, than Central Otago. And most importantly, Central Otago has most of its rain occurs during spring and summer, whereas all of the pruning in all three of these regions is done in winter, and Hawke's Bay and Marlborough have a winter dominant rainfall. So they're obviously at a higher risk already just by the timing of pruning. And we also have varietal differences. Marlborough is predominantly Sauvignon Blanc. Hawke's Bay is, has a lot of Cabernet Sauvignon, whereas Central Otago is Pinot dominated region, and that is known to be a lot less susceptible to the dieback than the other varieties. Pruning style is also important. Hawke's Bay is where most of the vines are pruned to a cordon, spur pruned, whereas we've got much more cane pruning occurring in the other two regions, and I'll talk a bit more about that further on as well. Water stress is something else that we have to deal with, particularly in Australia. So there's been a lot of talk about the effect of water stress on symptoms and the explosion of symptoms that we see in Australia and New Zealand over the last decade or two. So we set up a trial in the Riverland in South Australia. It's a very low rainfall area, and in that trial we had irrigation systems set up to provide 100% of what the vines needed, down to 12.5%. So quite low levels of irrigation. Excuse me. So as you can see here, the red line shows you the leaf water potential, and as the water stress increased, the leaf water potential increased. So you can see that the vines were definitely under stress. And then we've got both Utica dieback and Botrytis furei dieback pathogens. And what we can see is that this is again the distance that the pathogen the fungus has been able to move through that cane. And you can see in 100%, if we compare between well-watered vines and non-well-watered vines, if we look at the Botrytis furei pathogen, we don't see any significant change over that time. And in fact, for Utica, you see as the vines become more stressed, we actually find a slowing of the Utica fungus. So what we're actually seeing is the opposite of what we expected, and we're actually seeing that the vines under stress, the fungus is actually not able to move as quickly and progress as quickly. So going against what we actually originally thought. We had a student, Brittany Oswald from the University of Adelaide, do some extra work on this to try and understand why the differences. And she actually found that vines under low irrigation had much smaller xylem vessels and cane widths compared to those under full irrigation. So you can just imagine just physically the mycelium trying to get through these vessels, much easier in a well-watered vine that has good vigor. But she also found that a much higher lignin content. Now lignins are associated with disease resistance, and there was definitely a higher level of lignin content in those water stress vines than there was in the non-stress vines. So some really interesting results, which I guess go against what we thought, and so it's a really important finding that we've got from that work. So just moving to disease management, as Jose said, we're not doing anything really differently to what you're doing there in Canada. Cultural methods are by far, the most important to start with, and that's all around timing of pruning, and we'll talk a bit about that. Prevention through wound protection, we've spent literally 2 to 3 decades working on that here in Australia, and we've got some pretty nice results and lots of registrations. So so this is really the key, I think, to the future of managing trunk diseases in Australia and New Zealand. And finally, controlling the disease through remedial surgery. We have also, as Jose mentioned, done a lot of work there. So firstly, in terms of spore timing, we've been doing a lot of spore trapping, and you can see here we've set up traps across the southeastern part of the country, which is where the majority of vines are grown in Australia, and we've also had a couple of traps set up in New Zealand. I'm not going to, we have a lot of data, and I'm not going to spend too much time showing you the detail because it's probably not that important to you guys. But just to say that, you know, we have a real range of climates across between Australia and New Zealand, anything from warm subtropical climates that we get in New South Wales through the temperate regions in the southeast of Australia, and then very cool climate in New Zealand. The important thing to see is that we find spore release is pretty much all through the year. We don't have really a quiet period. Maybe during summer, on a very dry summer, you'll see some periods where there'll be no infections, but essentially, as long as you've got a rainfall, you have potential for spores to be produced. Certainly in the winter, we have the majority of spores are produced, particularly in the temperate climates. In the subtropical climates, we find that spring and summer seem to bring a lot more of the spores, and that's clearly where most of the rains come in the wet season. So, you know, this is really opened up our eyes to the amount of inoculum that's out there and the importance of protecting wounds all year round. And in the near future, we'll be starting to look at spring shoot thinning as a potential infection site, given the amount of spores that are in that that are detected in that time. So the next important story is the wound susceptibility. You know, we know that it varies between different regions and different climatic conditions. So we've been doing trials in lots of different areas. This particular graph is from a trial we ran in the Adelaide Hills, which is a warm, wet environment. And you can see here that with Udipa dieback, that these bars represent the amount of recovery. And I should just point out, Jose talked a lot about his inoculations and using 5,000 or 2,000 spores. We are actually down to using only 200 spores for our inoculations, because we find that the you know, going into the thousands has been just way too much disease pressure for us to get realistic results. And we feel that, you know, we understand that probably more like 10 spores might land on a wound of this size in reality. So we don't want to overpower these wounds too much, otherwise the information becomes a little less reliable. So given that we put 200 spores on there, we're still seeing very susceptible wounds when inoculated 1 day after pruning. And these pruning dates are early in winter, mid-winter, and then late winter, early spring. But what you can see here is that as we go through the season, we see a decrease, particularly in that late pruning, of susceptibility to those wounds immediately after pruning or in the first 24 hours. So this really shows there's some window there for you know, less susceptibility in this particular region in the Adelaide Hills, and we don't always see this in all regions in Australia. The really important thing that we see in all of our regions that we've been testing is that infection pretty much comes down to a negligible level, close to that to non-inoculated controls, within 14 days. So we believe that the susceptibility period really is the first 14 days, and obviously the first 7 days is the most critical. And when we're talking about protecting the wounds, we need to make sure that time is covered best. We see very similar results with the Botrytis dieback pathogen. We use Diplodia seriata in this case. And you can see here that, again, 14 days is pretty much, we're down to negligible levels of infection when we only put 200 spores on. So it really does tell us that 2 weeks of protection is the most important. Just incidentally, we had a PhD student, Bruce Henderson, recently do some really nice work on pruning wound surface area. He compared cane prune vines versus spur prune, as there's been an age-old question about which method is more, are the vines more susceptible, and there's been a lot of talk that cane prune vines, there's a lot less disease, so they're a lot less susceptible. So what he did is he did some assess some Shiraz vines in the Barossa Valley, and he compared these two types of pruning, and he found that there was 20 to 30% less wound surface area in your cane prune vines compared to those that were spur prune. So even though you got very small wounds on your spur prune vines, there's lots of them, and when you add them all up, and Bruce actually measured each of these wounds and on each of the different pruning styles, and worked out that there is definitely less surface area in these larger wounds than there is in this lots of smaller wounds. And he also found 38 to 50% less Udipa dieback in those cane prune vines, just like what we generally see. So what this summary of this is that fewer large wounds on cane prune vines for sure, and that amounts to less surface area than the many small wounds on the spur prune vines. But regardless of pruning method, I think it's really important to remember that limiting so obviously limiting your wound surface area will reduce your infection, but you still can get infection. And if you think about your cane prune vines, your wounds are closer to your trunk. Once they're infected, the infection is going to happen a lot quicker down into the trunk. So just because you're cane prune doesn't mean you don't manage the disease, but it will reduce the risk and make pruning wound protection a lot easier because you've got a smaller area to cover. Speaking of pruning wound protection, again, I'm not going to give you too much detail on our trials because there's at least 20 years of them. But essentially, to summarise, we have a lot of products that are registered, denoted by these little R symbols, and the little ticks tell us where we've trialled something and it's been very effective. So paints and paste by far are the best thing you can do. It provides a physical barrier, and in the case of these sort of products, you also get fungicidal action, and that'll prevent any infection getting through cracks that might appear, or if the sap flows and opens up the wound again, or if rain falls too quickly and washes off the paint. So that gives you a little bit of extra protection. But these products are all really amazingly good. So we obviously needed to move to something a bit more practical. So fungicides, water-based, liquid formulation fungicides was the way to go to see if we could spray them on. I'll talk a bit more about that in a minute. We have now 3 different active ingredients and a number of different products that are registered in both Australia and New Zealand. And we also found pyraclostrobin to be effective, but we're yet to see any registrations. But as you can see, there's quite a nice range of registered products there that are available in both countries. And in terms of organic options, we're probably a little thinner on the ground with that. We do have a Trichoderma species Vinevax product that's registered in both countries. Although recent trials with those have shown them to be more or less unreliable, and these things really rely on the right conditions to flourish. There is a product that's been registered in New Zealand called Knockblock, and it's really a paste, and it's based on a pine tar resin, and that's been working quite well for them as well. And we've also used a garlic extract, just simply, squeezing the juice out of garlic and applying that to wounds. We've shown that to be quite effective. This is an area where we are lacking, and working with Jose, collaborating with Jose is a really beneficial thing, because there's a lot of work that they've been doing in their group, which we hope to, in our next phase of research, start to take on some of this more biocontrol strategies to build this bottom end of this table for our industries. So spray application has been something we've also done a lot of trials on to I guess, show that it is effective to use your vineyard sprayers to apply the pruning wound protectants. Without showing you all the details, we've tested lots of different units. Obviously, recycle units are a great asset, because you're limiting the loss of the product or off-target use. But what's really important to understand is your coverage, and using water sensitive papers or some other method to look at your coverage, you really need to make sure your wounds are getting good coverage, because as you can see by this graph, the greater the coverage, if you're using a fungicide that's effective, the better your disease control. So it's all about coverage, and if you can do that, you know, spray application is a very effective method. And just as an aside, we've also done some work on spray adjuvants to see if the addition of those adjuvants does help the coverage. But unfortunately, we saw no difference between the use of those adjuvants and not using them. And this is mainly because a wound is very much like a sponge, and when a water droplet hits it, it pretty much soaks in, as opposed to a leaf where you have a waxy cuticle and the droplets tend to want to just bounce off and leave the leaf. So adjuvants are not really necessary in this case. Critical timing for application. Well, this is really important, and especially when you're applying with sprayers in winter, you know, trying to get the sprayers into the vineyard in wet weather. And just you know, the work health safety of pruners in the vineyard. So what we did is looked at the curative properties of the 3 different fungicides that we've got in play. We looked at inoculating the pathogen on the day of pruning. And then you can see here, there's your control infection. So we looked at about 60% infection when we do that. And then we come along on day 1, 3, or 6 and apply the fungicides and have a look at how much reduction in infection we see. And you can see here with this graph, and this is a bit of a summary graph of both Urbez and Botrytis pathogens that we've tested. We've got very similar results. But what you see is a really great reduction in infection in day 1, similar in day 2, and really even in day 3. Compared to that inoculated control, we really saw great control right out to 6 days. So we recommend now to growers that you've got up to 6 or 7 days to get your sprays on. And that will also increase the window of protection, because we also looked at the preventative properties of those fungicides. So that is that we applied the fungicides on the day of pruning, and then we came along and introduced the pathogen a day later, 6 days later, and 14 days later. So it really tells you how long is the fungicide still active. And what you can see from this graph is, compared to our non-treated controls, we got really good control all the way out to 14 days for some of those products. So what it tells us is we've got a week to get the product, we've got a week to get the product on, and we've got 2 weeks of control once that product's on. And if you think back to the susceptibility period being about 2 weeks, the critical susceptibility period, we feel that we've got that covered quite nicely in that timing. So a little bit of evidence, you know, it takes years and years to get to a point where you've got growers using methods for, you know, applying pruning wound protection for many years in a row. And we're now starting to see early evidence of that. In New Zealand, we saw from one of our surveys of 600 blocks around the 2 major regions of Hawke's Bay and Marlborough. We saw that typical graph of, you know, 10 to 15 to 20 years, real big increase in incidence, up to 90%. But when we just removed a group of 100 blocks which had protection of large wounds for the last 12 years, and then all wounds protected for at least the last 5 years, we can see here that we saw no increase in incidence. We saw a cap of 20% incidence across to 20-year-old vines. Now, this is really, really promising results, because you've got to remember a lot of these blocks were only started to be treated, you know, in the first, you know, around 5 to 10 years of age. So there was still that window of opportunity for infection, which is probably what this 20% is related to. So, you know, we feel that getting that protection from year 0 is going to make even more difference. And then when we look at some results of an Australian survey that we did, we're able to extract the data to I guess, further provide evidence about early wound protection. And what this graph here shows you is your dieback incidence in a number of different blocks that have been that have had wound protection applied for anything, or for either no protection or 1 to up to 11 years in a row since the protection started. So what I've done here is these blue lines represent the time, the years going backwards that any of these individual blocks had been sprayed. And if you look at the pattern, which is denoted by this red box, the time or the window where there was no wound protection, as it increases, we see an increase in the incidence of dieback in those vineyards. So those vineyards that have been treated from very early on are in the best position, still under 20% infection. So this is again more evidence to say, wound protection needs to start from the time you plant your vines, not once you start seeing symptoms at 5 to 10 years of age. So this has really changed the attitude of many growers in Australia, and we are seeing quite a quick increase in the application of wound protection. So moving to remedial surgery, Jose has just explained it. He's even shown this graphic. The key is to try and cut down to get past infection. I know that, in Jose's trials or in Jose's work, that it was very hard to get below infection. The infection seemed to go to the ground. That's obviously makes it very difficult, but it still may have potential and has got potential to extend the life of the vineyard. But obviously, if you can get right past the infection, you really could increase the life of that vineyard for a long time. So just as an example, a couple of vineyards that we went through and we cut vines at either low, mid or high on the trunk. We found when we did that, in the low cut vines, we only had 20% of the cross section still with staining. And then if you cut high, obviously a lot higher proportion of the cross section still had staining. So when we came back 6 or 7 years later and assessed particularly for Utica dieback in this case, you can see a similar pattern where only 20% of the vines had developed the symptoms of Utica dieback, and you can see that closely relates to the 20% of the stumps that still had infection in them. And as the cut gets higher and the incidence of infection gets high, you can see that those vines are all infected and reinfected after 6 or 7 years. So not such a successful way to manage the vines, cutting them up high with lots of infection. When we do remedial surgery, one of the problems that we have, particularly as we are predominantly owned rooted in Australia, the low cut vines, especially when they're greater than 20 years of age, they tend to be a bit stubborn, some varieties, Cabernet and Shiraz particularly, at producing water shoots. Pinot and Malbec really good at producing water shoots, so great contenders for remedial work. But Cabernet and Shiraz, we need to intervene in some way because 60% of the stumps coming back is not viable at all. We did find also in grafted vines that we have a lot of problems getting water shoots, and this is something that you guys in Canada would experience more as you have a lot more grafted vines. And as you can see here, we looked at Semillon as an example. We had some own rooted Semillon, and we had no problems low, mid or high cut with the production of water shoots in the sign. But in the grafted vines, you can see we had a lot of problems, especially in the low cuts. We either got no shoots or shoots just coming out of the rootstock and very little coming from the scion. As you cut higher and higher, you did get more scion, induced water shoots, but of course, you have the issue once you cut high of more infection still in the trunk, and you still have quite a lot of no growth. So there's some issues there that we decided to follow. When I show those graphs in Australia, I have a lot of growers tell me that they have all sorts of methods that they use to induce water shoots when they do this work, methods they swear by. And so we decided to test those in a scientific, rigorous experiment. We grabbed a hammer and hit the side of a trunk. We used a wire brush to scrape and open a little bit of a wound on the side of the trunk. We used a tomahawk to mark an X, and we also applied a hormone Dormex. And all of these things, are things that growers have been using to try and stimulate the growth of water shoots once they cut these stumps. So what we found in this experiment was essentially, and I should have added, and it's pretty obvious from the graphics here, that the wound, the applications all happened within a white circle. And if you look at this graph here, the blue bars down the bottom, they represent the amount of shoots that came out from within that circle in each of those treatments, including the control. And you can see here, a very small number of shoots came out from those circles, and they were, you know, not significantly different than each other. Most of the water shoots came out from anywhere else on that stump. And again, not really any difference between the treatments. So this really showed us that none of these treatments, you know, significantly made a difference to water shoot production. So we continued this trial because we actually looked at grafting as well in that first experiment. And we continued to work with this for 4 or 5 years. We have another year that I haven't got marked here. But what we did is each year following this trial, we actually just did cuts in winter and spring to see if there's any difference between the water shoot production when you cut early in winter or if you cut in spring. And then we came along and we grafted any of the stumps that had no shoots to see if we could increase the level of water shoot production. And so you can see in 2017, we don't see a significant difference, even though there's a slight difference in winter. It wasn't significant between just the natural water shoot production. But what we did see is when we grafted these, we did get an increase from 80 up to 93% or 87 up to 98%. We saw a 10%, at least a 10% increase in water shoot production, which was quite a significant amount. So we continued this. In 2018, we only did a spring cut. And then in 2019-20, we went back to doing both winter and spring cuts as we did in 2017, and then grafting. And as you can see, this continued on this trend of no real difference between cutting in winter and spring, but any stumps that didn't have water shoots, we grafted, and we saw up to 20% increase in water shoot production. So a real promising result. And essentially has led us to recommend to growers now to use grafting as a filler for those stumps that don't work. Obviously, there's always going to be a number of stumps that have died completely, and no form of grafting would be able to make that up. But you can imagine 90% of trunks with shoots is much easier to manage than 60% that we saw in the graph earlier. And just to finish off, I just want to talk quickly about some economics work that we did in New Zealand, or more importantly, Greg McCarthy did for us in New Zealand. So what this graph is trying to do is trying to help explain the cost differences between different remedial and preventative responses. So it looks at the relative annualized NPV future cost by doing any of these things, going forward at whatever stage you're at the time. So the way it works, if we take the graphic, if you consider your vineyard if you go through and you assess your vineyard and you've got 30% of vines have got some amount of infection, well, this grey line here tells you if you do nothing from here on, the fact that you've got that much disease will mean $4,500 per annum per hectare going forward by doing nothing. So if you then choose to just spray annually, which will reduce your cost going forward by $500, obviously you've already got infected vines, so those vines are going to continue to die. You're just going to stop any new infections. However, if you take the next step and you go, all right, let's remove and replace the symptomatic vines and then spray annually, you can then see another $1,000 saved per hectare per year going forward. And then even further, if we then consider, rather than removing and replacing, let's rework and graft symptomatic vines and then spray annually. So you can see we brought our potential cost going forward down from $2,000, which is obviously very significant in terms of the long-term sustainability. So reworking and regrafting obviously takes into account that you have a lot less loss. You have a few years extra yield that you wouldn't have got if you remove and replace the vines. And obviously the costs of reworking are a lot cheaper than replacing vines. If we take another scenario, more infection in the vine, we can see that we're looking at $8,000 per hectare each year going forward. So, you know, it's starting to become very expensive. If we just spray, we reduce that by 500. And you can see now in much more, when you've got much more infection, by removing and replacing and then even reworking, you're down to less than half of the cost going forward. So you've really saved a lot of money by applying these strategies. And just as a final scenario, a vineyard which is quite heavily infected, 80% infection, you know, there's really no point to think about all these other options, because by this point you're in a better position to just rework and regraft all the vines and then go through and spray annually. So wholesale reworking was really the best thing you can do by the time you get to this amount of infection. So these graphs really just help us, I guess, visualize the costs that would be incurred and what can be saved by applying a lot of these remedial and preventative responses that we have developed. So just to talk about resources, Jose showed this graphic. This is a paper that we wrote together with our other colleague, David Gramahay, who you heard from last year. It's apparently just been announced that it's the highest referenced journal in plant disease for the last 5 years. So it's become quite a commonly read and followed guide for scientific management of trunk diseases. So that's a really good place to go looking. In Australia, we have an app which helps you assess your trunk disease manually. So that's something that's freely accessible. We also have been producing a lot of YouTube videos, not only myself sort of talking about the disease, but also grower experiences. And I think you can't get better than actually hearing about growers that try some of these things and give some feedback on you know, the pros and cons and where it's left them. So all of these are on YouTube, available for you guys to watch if you want. Probably the easiest way is to go to the oneaustralia.com website. And we also have a best practice management guide, which gets updated every year or two. And that also covers off on all of the things that I've talked about today and lots more as well. So it's become quite a handy tool for the Australian and New Zealand grape growers. Just to finally thank, you know, this isn't just my work over the last 20 or 30 years. It's actually, you know, I've had 2 research teams here, one here in Australia and one in New Zealand. So I just want to, I guess, acknowledge them and their hard work, which has, you know, presented us with all of these outputs. And obviously a lot of funding coming from Wine Australia, New Zealand Wine Growers and Brigado Research Institute. We work with a number of different groups in Australia and New Zealand. And of course, our collaboration with Jose and with this, you know, the European groups. And we're very heavily involved in the Council for Grapevine Trunk Diseases. With that, I thank you for your attention, and I guess we'll be opening to questions.
Perfect. Thank you so much, Mark. That was wonderful. I will turn it over to my colleague, Bill Armstrong here. We're a little tight on time for questions, but I see 4 in the Q&A box right now. So Bill, if you want to go ahead and just start reading through those one at a time, and we'll open it up to Jose and Mark.
Yes, I think this one is for Jose. What are the air humidity and temperature ranges required for Trichoderma to thrive in the field during application?
Yeah, that's a very good question, and that's we know from previous studies in other geographical regions that the relative humidity temperature may vary depending on the species of Trichoderma we have. Overall, most of the Trichoderma, even though, for example, the ones we found here in BC, which were we thought at the beginning they would be maybe more acclimated to a cold temperature for development and growth. Most of the Trichoderma, they have their optimum growth at 25 degrees. So that's one of the handicaps we have, maybe if we think on applications under our weather conditions. However, very interestingly, we have done some work on spore germination, and we have found that the Trichoderma spores of some of these species can stay on the pruning wound, I would say, latent for a time, but they are able to germinate with temperatures as low as 10 degrees. So I think it varies, and that's what I mentioned. We need to do further work here to see how now, knowing the species we have, that they produce a very good control, how we can improve this application, how early in our season we can go, thinking we may have to do applications, maybe February, March, where would be very, very, at the edge of getting those temperatures for optimum germination. So this is some of the work that we still need to do.
The second question is for both. Are differences in susceptibility, duration, a function of strains of pathogens in BC versus Australia and New Zealand, or environment? Are differences in susceptibility, duration, a function of strains of pathogens in BC versus Australia, or environment?
You want to start, Mark?
Well, I can if you like. Look, it's probably a number of factors. The differences that you can see, particularly between the work that Jose and I have been doing. Of course, the pathogens are different strains. The climate is different, so the pathogens grow differently in those climates. But as I pointed out, just the concentration of spores that we put in our trials will make a huge difference. And over the years, we've used all different levels of concentrations and got very different results. So what's really important is to understand that what number of spores, landing on a wound is actually realistic. It's actually something we're planning to do in the next few years to get a much better grip of that, because we're basing what we know of 10 spores landing on a wound from 50-year-old research that was done on apricots. So it potentially is different now and on grapes. So, but the reality is it's much lower than what we test. So that's probably the key to the differences that we see between trials across, between New Zealand, between Australia and Canada.
Yeah, I agree with Mark. I think one of the important things is, for example, within Botrytis area, we have to think about there are many different species involved in the disease, and we have observed that even the different species show different virulence in the plant. So we have a species, with much lower virulence than others. So that's a factor to take into consideration. And yeah, I don't think we exactly know how many spores a wound can receive per year in natural conditions. There's some research that maybe, Mark, we have to get together to endeavor, what's really, the number of spores a vineyard can get in a certain season. But definitely, we have a much higher number of spores. We want also, of course, our controls to be significant. But at the same time, as I mentioned, that probably underestimates some of the efficacy of these products that they have. So we are planning to now that we have the whole system very well developed, to go to number of spores closer than Mark has, to get closer to a reality of what natural conditions are.
But just to add to that, Jose, you even using those higher disease pressures to see such good results, particularly with your biocontrols, is really builds confidence, because if it works at that high disease pressure, it's not a problem at anything lower.
Exactly.
For Mark, are all registered fungicides equally effective on both Utupa and Botrytis?
Yes. So all of the products that we have registered, a lot of them were registered originally just for Utupa, but we've tested all of them now for Botrytis, and so anything that's registered, we are recommended now for both Utupa and Botrytis.
And last question, does snow activate spore release?
That's a very good question. So we have some data from studies done in New York and Michigan on the 80s on Utypa, that actually they observe a spore release during some parts at the end of the winter, where it doesn't really clear those studies if the spore release were, due to snow precipitation. It may happen, and we actually have captured spores in the winter here in BC, very, very minimum. Contrary to, for example, in Australia, our number of spores during the winter are very, very low. One of the things I think more than the precipitation snow is just the temperature. I think our temperature conditions are, for many times during that winter period below zero, which even under precipitation that you would think they would enhance spore production and dissemination, I think it has something more to do with the temperature than the precipitation at that time. So we don't see these high numbers in spores. So I don't really know. I mean, the question, the sad question to the snow is, I don't really know if the snow, good, but I think it's more the temperature is so low that's why we don't see those conditions. Also, in the case of, for example, Wishlist, in many occasions, you will need this heat of the rain droplets into the fruiting bodies contain the spores to disseminate those spores, which I believe that probably snow will not have that pressure and that effectiveness of disperse those spores. But I think it's more related to the temperature condition.
Just one last comment. Allegro is now registered on grapes in Canada. Another project for José.
Yeah. I had to say that some of the issues we have, for example, during my time working in California, where we have way more products registered, same as Mark in Australia, on trunk diseases. I think most of the products coming to Canada, they come registration for, the main issues as powdery mildew, downy mildew, botrytis. Main issues. I said that it's hard to start registration for a new product in Canada with the amount of acres we have, and some of these companies, they probably focus the market in other regions where the number of acreage is much, much higher than we have in Canada. But I think fortunately, thanks to our work and to our data generation, I'm very confident that we are gonna soon have some products for trunk diseases.
Yeah, I should also add to that, Jose, that, we have now, as you see, a lot of registered products, but our work started almost, probably 25 years ago in screening products. So we're probably almost 20 years ahead of Jose's work. It does take a long time to get the data necessary and then get your companies to register these products. It's not an easy task. So, you need to be patient, and but the rewards will come, and they'll come, before it becomes a big problem for the industry in Canada.
Perfect. Thank you, everybody, for the Q&A, for submitting your questions. I just have a
Sorry, Ethan.
Oh, yeah.
If I just have one second, because I just realized on me rushing, to get time for Mark to get the presentation. I know we are out of time. I forgot the most important slide. I don't know if you guys can see that. But saying, as Mark mentioned, this is not the work of one person. We have a huge team at AAFC, as well as collaborators we have, and the funding. So I just wanted to make sure that I acknowledge, the funding that allow us to do this work, and all the people behind this work. Is not just me. So I just realized that before when I finished my presentation. So just wanted to mention that.
No, that's all good. I think that's very valuable, and I'm sure everyone who worked with you appreciates that. So thank you for that. I just have a couple final, I don't realize I started at the beginning here. Just a couple final wrap-up slides to share. Okay, perfect. So just wanted to thank everyone once again for being here, and obviously special thanks to our presenters, Jose and Mark. This was very valuable, I hope, to everybody who participated. I certainly learned a heck of a lot in the last hour and a half or so here. This is just a brief slide I have to show to legally ensure that we can post this webinar to our website. So I'll just let that display for a second there. And then this last slide here for us, you can join us again throughout 2022 for the other webinars that CGCN will be hosting. We have several coming throughout the spring and summer. focused on topics such as crown gall, tissue culture, Pinot Gris virus, Syrah decline, and several cluster research updates where you can learn more about the progress that's been made on the cluster projects to date. Also, be sure to follow CGCN on Facebook and Twitter. You can subscribe to our email newsletter, as well as check our website for regular updates if you want to be informed about the future webinar sessions. Links to register for the next one in February 17th, the first cluster research update. Those links will be available shortly. As well as I mentioned, this webinar has been recorded and it will be posted to the CGCN websites, both the English and French websites, if you ever want to revisit this video or send it to someone who wasn't able to attend today. Last quick thing, when you exit the webinar or when I end the session, you'll be asked to complete a brief survey where you can share your thoughts on how today's webinar went. We'd really appreciate it if you could fill that out just to give us some feedback that'll help us improve our future webinars. So once again, just a big thank you to our guest speakers, Mark and Jose. Thank you so much for being here. Mark especially got up at the crack of dawn for this, so that is much appreciated. Thanks to Bill as well for moderating and being my additional CGCN representation here today, so I appreciate that. Thank you to everyone who attended, and we really hope to see you at some of our future webinars in 2022. So thank you very much and enjoy the rest of your day, everybody.
Thank you very much.
See you later.